So then I think it's right to get started. First of all, welcome to all of you to the session of physical vectors. And the special focus of the meeting is immunotherapy. Therefore, we have three excellent speakers. I'm really happy on this. And the session will be led by my colleague Amir Khan. He's sitting in the first row, and myself, Ernst Wagner. We will step down from the podium and we'll get up 3 minutes before actually the end of the talk. And it's now my great pleasure to introduce the first speaker. It's Dr. Lisa Villanueva, and she will talk about therapeutic application mechanism of zone operation.
Thank you very much to the organizers for inviting me to give this talk. I wonder if we could have the slideshow format. Great, thank you. So I was asked to talk about some of the work that we're doing at the University of Pittsburgh on sonoporation. So this is not actually an immunotherapy talk, but hopefully it will provide some relevance to those interested in immunotherapy. So by way of introduction, ultrasound, as you know, is really a physical phenomenon comprised of compression and rarefaction waves, whereby you have high pressure, low pressure, high pressure, low pressure in succession. And ultrasound contrast agents are microbubbles or gas-filled microspheres that, because they're gas-filled, are compressible. And in the presence of an ultrasound field, they manifest behaviors supposed to be in this movie. Let's see if this plays. Whereby they oscillate, they vibrate in response to ultrasound. This is a movie supposed to play that captures microbubble acoustic behaviors at 25 million frames per second. Using a special camera that we have. And these microbubbles transit through the microcirculation as shown here in this intravital microscopy where they're fluorescently labeled. Now when they oscillate in the presence of an ultrasound field, they become ultrasound emitters themselves. So the transducer both delivers the ultrasound and then receives the ultrasound emitted by the microbubbles, and this results in a contrast effect. This is something that's used in clinical cardiology for FDA-approved indication in patients who have an echocardiogram that's technically limited, such as shown here, where you can barely see the endocardial surface of the heart and you can't evaluate wall motion of the heart. And when you inject these contrast agents or these microbubbles, they light up the blood pool and are able you to see the walls of the heart more clearly. For therapeutic applications of ultrasound contrast agents, we capitalize on the same behavior, but we use it for a therapeutic application. So again, these are microbubbles that are insonified at a low acoustic pressure and then at a higher acoustic pressure. And when that happens, you can have either stable cavitation or inertial cavitation, where you have very bizarre oscillations of the microbubbles resulting in cavitation. And this results in energy release that can be harnessed for therapeutic use. The concept of it is actually illustrated in the life of a snapping shrimp, which divers can actually hear as popping sounds when these snapping shrimp clasp their claws. Again, this movie is not playing, I'm not sure why. But essentially, when these claws snap, there's a violent, high-energy cavitation event whereby a bubble is released, and what happens is the shrimp uses that energy to temporarily paralyze its prey, and when it does that, the prey gets paralyzed, and then they get the prey. And so the concept is that in nature, acoustic cavitation is utilized as an energy delivery method with biological effects. Okay, there it goes. So this is the shrimp. And it snaps its claws and it paralyzes the prey temporarily. And so this cavitation event is, in nature, is what we use for therapeutic applications of ultrasound contrast agents. So we use microbubbles as therapeutic vectors. So we use ultrasound-targeted microbubble destruction, which UTMD, whereby these microbubbles act as flexible non-viral structures that can be designed to carry a payload to target tissue. As I mentioned, the microbubbles oscillate or they burst in an acoustic field, and this oscillation itself increases membrane permeability, such as the endothelial permeability, and the microbubble destruction releases the payload at the site of ultrasound delivery, where the membrane permeability is enhanced. So we use externally applied ultrasound, such as available on diagnostic echo machines, and the spatial targeting is achieved by navigation of the ultrasound beam. So the delivery occurs only where the ultrasound is delivered. And we can do simultaneous imaging to allow real-time confirmation. So it's a theranostic approach. And the concept is that this method will overcome some of the barriers to delivery of nucleic acid. So we have a nucleic acid-loaded microbubble that is protected from digestion by serum nucleases because of the PEG brush on the surface of the microbubble. The microbubble circulates and is able to transgress the endothelial barrier because the rupture event causes transient permeability of the endothelial lining. And then in addition, the destruction event causes a pore to form, or sonoporation, which allows the nucleic acid to enter the cytoplasm through an endocytosis-independent transit, which allows for more efficient delivery in the sense that it does not have to undergo lysosomal escape. And that's really the approach that we've been using. And this is a schematic of how this works. So this is a rat heart, and you can see here, this is ultrasound contrast opacifying the myocardium itself. So you can see where these microbubbles are going. And then because of their vibrations, they can cause microjets or microstreaming that can induce pores. And I'll show later in some pores that are induced in endothelial cells. Now, importantly, microbubbles outside the ultrasound field retain the payload so that you have no off-target uptake. So the targeting is achieved by navigation of the ultrasound beam, and also you can see what you're doing by virtue of the imaging capabilities of the ultrasound modality. So we've used this to treat head and neck cancer, which is the most common head and neck malignancy. There are 5,000 new cases annually. Its most common presentation is late, and it has dismal prognosis. It's a very expensive disease, and various approaches have now improved mortality and results in much morbidity. And fortunately, there have been pathways and proteins involved in the oncogenesis that can lead to targeted molecular therapeutics, but there are limitations in packaging and delivery systems, and this is where our delivery approach can be advantageous. So we initially targeted the EGFR pathway, the epidermal growth factor receptor pathway, which is very involved in multiple cancers, such as head and neck cancer, in the proliferation, invasion, angiogenesis, metastatic potential, and its resistance of tumor cells to apoptosis. And what we did is we loaded a lipid microbubble, which is a perfluorobutane gas-filled 2 to 3 micron-sized microsphere with EGFR directed, siRNA directed against EGFR. We have a tumor model of SEC7. It's a murine squamous cell carcinoma model on the dorsal surface of the mouse. And we use a clinically available ultrasound machine, and this is just still frames showing how we do this. So this is the tumor itself, and this is before microbubbles we use a contrast-specific imaging modality, and this is before microbubbles have perfused the tumor. We continuously infuse the microbubbles intravenously, and the microbubbles perfuse the tumor. We then increase the energy very briefly and break the bubbles to deliver, and this is what it looks like right after breaking. And then we wait again to perfuse, which is just a few seconds, and we do this iteratively, perfuse, break, over the course of about 20 minutes. This is examples from 2 mice, and these are the series of mice. This is from a microbubble that's loaded with a control siRNA, and these are 3D-dimensional ultrasound images from after delivery of an EGFR siRNA on a microbubble. So as you can see here, this is day 1 and this is day 7, that there's progressive growth of the of the tumor that was treated with ultrasound and a microbubble loaded with a control siRNA. And here you see the results of the tumor growth in an EGFR siRNA microbubble-treated animal. As you can see here, the doubling time is significantly prolonged in our treatment group compared to the controls, meaning no siRNA and ultrasound, control siRNA and ultrasound, and this is EGFR siRNA without ultrasound. And this is a compilation of the growth curves. So we were encouraged by this showing that our technology is able to inhibit tumor growth. And when you look at survival curves, you can see that this is our treatment group here, and these are the control groups, that there's a improvement in survival in these mice as measured by the tumor growth less than 8-fold, that is, at which point we have to sack the mice because the tumors are so large. We show that in cultured SEC7 cells that there is a decrease in proliferation in vitro after we deliver the siRNA via the microbubble ultrasound technology. And these are histologic immunofluorescently labeled specimens for EGFR. And you can see here EGFR expression in the tumor. This is her staining for nuclei, and the green is immunofluorescence. And here you can see a down regulation of EGFR expression associated with an increase in apoptosis compared to the control. And here is protein expression of EGFR, and this is our treatment group, EGFR plus ultrasound. You can see a down regulation of EGFR compared to the siRNA without ultrasound, and then to the EGFR nonsense siRNA with ultrasound. We then collaborated with the lab of Jennifer Grandis at our Cancer Institute, and she's been working with STAT3 in head and neck cancer. Now, STAT3 is a transcription factor that's hyperactivated or overexpressed in numerous cancers, including head and neck squamous cell carcinoma. And this contributes, this is a transcription factor that translocates to the nucleus and facilitates the transcription of target genes which mediate proliferation, survival, migration, angiogenesis, immune evasion, and ultimately tumor metastasis. And this is associated with resistance to therapy and reduced survival, this overexpression of STAT3, and therefore is a potential therapeutic target. So Dr. Grandis designed this transcription factor decoy, which is an oligonucleotide sequence. It's recognized and bound by the transcription factor, and it results in competitive inhibition of binding to the corresponding genomic DNA. This is a double-stranded transcription factor decoy that we then attach to one of our lipid microbubbles. And as with the EGFR siRNA, these are some examples. This is the decoy treated animal showing a delay in the growth of the tumors, although you can see that it starts to accelerate again after the treatment. We give 2 treatments on day 1 and day 4, I believe, but then you see that unfortunately the effect, at least with this regime, is not sustained. But importantly, because this is non-invasive, this is just intravenous, this could be repetitively given over the course of any number of days. We're just limited in doing this in mice because we have to put them under anesthesia to do this, but in a human being, this can be done in a conscious ambulatory individual, just like an echocardiogram can be done. So you can see here that, that with this delivery method, as with the EGFR siRNA, we have downregulation of target genes after delivery of the STAT3 decoy. And we also did some radiolabeling studies of the decoy on the microbubble and showed that there is preferential accumulation of the decoy in the tumor itself as a result of our targeting strategy, the targeting strategy being the ultrasound beam itself. So I'm going to switch now, having shown that the method actually works. We're really more interested now in how does it work, what are the biophysics of sonoporation. For one thing, it does remain less efficient than viral methods. Despite its advantages, it is less efficient than viral methods. So we sought to study the biophysics associated with the microbubble cell interaction. And in particular, we believe our paradigm is that there are issues with the microbubble physics themselves, the oscillations, the membrane physics, what happens to the membrane, and also what happens to cell-to-cell communications when microbubbles vibrate. And so a postdoc in my laboratory undertook this study, which really was a unique approach that couples imaging methods that span the intrinsic timescales of sonoporation. And by that I mean microbubble oscillations are in the microsecond timescale, and that's a microbubble physics part. The diffusion physics of the macromolecules or the drugs is in the millisecond to minutes timescale. And the plasma membrane biophysics is really in the seconds to many minute timescales. So we sought to combine all of these timescales in a way that we could understand the relationship between the microbubble behaviors and the resulting sonoporation events. And the way we did this was to couple two camera systems. So we have our so-called UPMC cam. This is a camera, it's an ultra-high-speed imaging system that enables us to image at up to 25 million frames per second, which is the frame rate that we need to resolve the temporal sphere of microbubble oscillations, because we're sonifying the microbubbles at megahertz. So we have to be able to image at megahertz frame rates to see the oscillations. And then the fluorescent events are where we have so we have here an optocell in which there are endothelial cells, and then we have the microbubbles here that are by flotation up against the endothelial cells. And then we have an ultrasound transducer here, and this is a heating coil to keep the cells heated. And we have a flash lamp here, and we have, through this objective, through a series of mirrors, we're able to direct some of the light to our ultrafast imaging system so that we can image the microbubble oscillations. In addition, we have a fluorescent lamp and then a CCD camera that over a longer time frame is able to simultaneously image the fluorescence events. And in this particular instance, we're looking at propydium iodide, which is normally impermeant to cells, but in the presence of sonoporation will enter cells and become fluorescent after binding to nucleic acids. So what we did was we get some still frame images initially, and then we deliver the ultrasound, which we have here as a variety of different ultrasound conditions, whereby we vary the pressures from 0.1 to 0.8 megapascals. We use 0.5 megahertz, 1 megahertz, 2 megahertz frequencies. We deliver a single ultrasound burst. And during that time, we can image the microbubble oscillating, and then we have the fluorescence imaging at a slower frame rate to look at what's going on with the propydium iodide. So this is what a typical example would look like, and unfortunately, this movie doesn't play this one here, but this is a microbubble oscillating as similar to what I've already shown. And you can see here the propydium iodide. This is the whole cell here, and the microbubble, this here, is up here relative to the cell. And you can see here when the ultrasound turns on, propydium iodide enters the cytoplasm and then goes to the nucleus over the course of 70 seconds. And this is the example of the kind of data we can obtain from this type of recording. So we have here the microbubbles oscillating cyclically in response to the ultrasound, so we can get radius versus time curves. This is auto-advancing. Radius versus time curves, and then we can do FF transformation and look at the power spectrum associated with this, so you can see the fundamental frequency in the second and third harmonic, so we can look at the frequency content of our oscillations, and then we can take regions of interest over the cytoplasm and over the nucleus and map it as a function of time after the ultrasound event. So here it's in the cytoplasm, it peaks, and then as it enters the nucleus, it decreases in the cytoplasm and reaches a steady state in the cytoplasm. This is the nucleus, also reaches a steady state, presumably because the pore at this point has sealed, so no more propydium iodide has entered. So these are some data for all the microbubbles analyzed. There's something like 200 microbubble cell interactions at 0.5 megahertz, 1 megahertz frequency, and 2 megahertz frequency. And this is the initial radius of the microbubble, and this is the change in the radius size. And red represents cells that took up propylium iodide so that they were sonoporated, and an open circle is no sonoporation, so no propylium iodide. And interestingly, what we found that regardless of the radius, that there seemed to be a threshold above which, of absolute excursion of the microbubble, above which sonoporation would occur, and below which sonoporation would not occur. And this was approximately frequency dependent. And so again, there seemed to be a threshold. Now, if you take this absolute excursion of the microbubbles, that is maximum radius versus minimum radius, you can convert that to a shear rate. And we find that at these different frequencies, there is a threshold for shear rate above which the microbubble will cause membrane disruption. And so you can have any number of acoustic parameters, but if a threshold shear rate is achieved, the membrane will break and a pore will form. And then what we found that is if this is now the maximum shear stress, and this is the maximum cytoplasmic propylium iodide signal, that after you reach the threshold shear stress at any given frequency, that the higher the shear stress, the more propylium iodide. I don't know why this is auto-forwarding. The more propylium iodide is taken up. So there's in a direct linear correlation. It's as though the higher the shear stress, the larger the pore and the more prolonged pore opening, so that there's more propylium iodide that's taken in. And so, again, putting up this slide where we've now looked at the microbubble physics and the macromolecule diffusion physics, and now we sought to look at the plasma membrane biophysics. And to do this, we performed confocal microscopy of real-time sonoporation. And in this particular model, we had a confocal scope, and we had the HUVEC, these are human umbilical vein endothelial cells in the microbubbles. We provide the ultrasound through a coupling cone here, and we looked at the permeability again using propylium iodide, the plasma membrane using a FAP label. I won't show the data on our intracellular calcium data, but also viability of these cells using calcium. And in this particular instance, we went with a 1 megahertz single burst 8-cycle ultrasound pulse. These are successive frames from our confocal microscopy, and you can see here that these are 1, 2, 3, 4, 5 cells, and this is 0 minutes all the way up to approximately half an hour. And so you can see here that this is where the microbubble was, somewhere here. And as soon as the ultrasound is delivered, you see this red appear, and you also see a hole that forms here. That is a pore. And what's interesting to see is that there is this wavefront of propylium iodide that enters the cell and migrates to the nucleus. And you can see initially that there's a trailing edge here of propylium iodide right next to the pore, but no more propylium iodide is entering after about between 4 and 5 minutes, even though there is still the appearance of a hole in the cell. There's no more propylium iodide going in, and it's all heading towards the nucleus. So from this, we surmise that the cell still has a hole in it from basal to apical side, but that the inside of the pore, the inside border of the pore is sealing so that no propylium iodide is actually entering the cell any longer. And the other interesting, and I'll show the movie of this in a second, the other interesting thing about this event is that you can see that as this hole is forming, there's also a disruption of the junctions between these endothelial cells. They become very dynamic. And this corresponds most likely to the contention that the event also causes a transient increase in endothelial cell permeability that facilitates the exit of nucleic acids. So what we're seeing here is both a pore as well as the dynamic nature of the junctions between the endothelial cells. So this is now the movie of this. Hopefully it'll play. So you can see here a fenestrated, initially it's a fenestrated pore that then ultimately seals. This is a 30-minute recording, so by 30 minutes the pore is gone. But note that the propydium iodide accumulation is only over the course of a few minutes, so that the pore itself has sealed. And then again, you can see these dynamic changes between the cells themselves as the pore is being repaired. You see changes in the junctions between the endothelial cells. This is in three-dimensional, where you can see more clearly that there's this trailing edge of propydium iodide and no further entry of propydium iodide after just a few minutes, despite the fact that there appears to still be a hole in the cell, again suggesting that the hole is sealing from the inner border of the pore. And then there's a more delayed healing of the hole itself. And you can see here again, if you quantify that this is the pore size, and this is the propydium iodide. So propydium iodide has plateaued despite the fact that the pore is still there, although it is diminishing its size. So it's two different time frames. And this is just mapping the gaps in between the cells here, showing as a pore is healing, you have increasing sizes and increase and decreasing dynamic changes in the gaps between the cells. Now here, after doing all this, we wanted to confirm that our cells were actually still alive, and so this is a calcium staining of these same cells at 40 minutes, showing that these cells are still viable. The other experiment that we did was, in this particular example, the bubble was still there after the initial sonoporation event here, where you see the propydium iodide. And then what we did was we hit it because we thought, well, maybe it's just saturated and that's why it's plateauing. But then we hit it again, and you can see more propydium iodide come in, confirming that the initial event caused poration, there was sealing, and then another ultrasound pulse caused another poration event and further entry of propydium iodide. So to conclude, our studies have shown that sonoporation can be used to therapeutically deliver nucleic acids, and we've shown this in head and neck cancer in the examples I've shown here, but we've also begun to develop microRNA delivery in cardiac disease, which is, I'm a cardiologist, so that's more my area, and so we've been doing some exploratory work in delivering microRNAs and antimeres to mediate left ventricular hypertrophy in a hypertrophy model and fibrosis in an angiotensin II model with really interesting results. And again, this is non-invasive, it's targeted, and it's image-guided, and you can keep giving it on successive days, and it's non-immunogenic because the bubbles themselves are physiologically inert. The ultrasound event, which is short-lived and in the kilopascal range, results in localized shear that creates cell membrane perforation and pores. And interestingly, it appears that the greater the shear stress, the greater the concentration of the uptake, so that we have the potential to regulate the poration event in a way that can be more suited for the given application. The membrane breaches that we saw conceal along the lateral area of the pore, that is on the inside of the pore. And so despite persistent opening in this XY plane, we have, and the propydium iodide influx ceases at 2 minutes, you still have this opening that persists for 10 minutes or even up, but sealing that occurs within 30 minutes. And we showed that treatment of the same cell twice with ultrasound yields further PI uptake, confirming that the initial event reseals, and then you can hit it again and it reseals again. And these poration events have bioeffects over different time scales. The direct increase in cell membrane permeability, which is a result of the hole itself forming, and what we can see with propydium iodide entering the cell, is on the level of 2 minutes, But the indirect effect, whereby there's an increase in membrane, presumably an increase in membrane permeability resulting from dynamic changes in the junctions between the cells, is something that can persist over the range of 30 minutes. So I just wanted to acknowledge the contributions of the people in my lab for this work. In particular, Brandon did the high-speed imaging work. He's funded by a grant from Quebec, and he did the confocal work. Jonathan Zukai and Andy Carson did the head and neck cancer work. Simon Watkins heads up our structural biology imaging center, provided the confocal facilities, and our technicians provided significant support for this work. Thank you for your attention.
Many thanks, Professor Villanueva, for your excellent presentation, which is now open for discussion.
That was great. I hadn't seen some of that high-speed imaging. Incredible. I just wanted to go back to the first part of your talk, which was this idea of intravascular infusion of the bubbles loaded with some payload and then targeted destruction. So I guess my question really centers on the bubbles that are not destroyed during this, because the target and the vascular space there comprises a tiny fraction of the overall vascular space, where the concentration of bubbles will be uniform across the entire intravascular volume. So what is the fate of those bubbles that you don't destroy, and what do you think the significance is of that vis-a-vis regulatory consideration?
Yeah, that's a good question. So, and we haven't done the systematic studies to look at that. What we have done is, well, if you look at all the work that's done for just lipid bubbles in general, there's 2 FDA-approved bubbles, Optisana and Definity. Most of those bubbles end up getting metabolized by the Kupffer cells in the liver, and the gas gets exhaled in the lungs. So we presume that that is what's happening with our lipid bubbles as well. As to what happens to their payload, that's unclear too. So I showed some of the studies on when we radio-labeled the STAT3 decoy. And we do find preferential accumulation in the tumor versus the surrounding tissue. However, we do see a significant amount of it in the urine, and also in the liver. But STAT3 is not hyperactivated in the liver, so we don't think that it will have bioeffects. But again, we don't know.
Thanks a lot.
Next question, please.
Beautiful talk and beautiful work. So my question actually continues what the previous question is that. So I see that when you use different size of bubbles during the ultrasound application exposure, that you don't usually larger microbubbles can cavitate much more violently, and they can induce more cavitation. But I don't really see that in your figure. That seems like all your bubbles has the same effect of taking this effect. So maybe at this lower pressure, acoustic pressure, maybe both inertial cavitation and stable cavitation both contributes to the uptake of the propionate iodide?
Very, very possibly. I think, well, first we did look at a range of pressures from 100 kilopascals all the way to
800.
Yeah, and then there's different sizes. I didn't show the graph, but if you look at if you use the high-speed imaging studies to determine whether this was stable cavitation or inertial cavitation, for our successful sonoporation events, about half of them were with stable cavitation, and about half of them were with inertial cavitation. So both behaviors do appear to contribute to sonoporation, and the governing principle appears to be the shear rate. not so much whether it's stable or inertial. It's what's the resulting shear rate that the cell membrane sees.
Right. So do you see actually increased propionate iodine? Because I didn't see, maybe you had a figure from like 100 to 800. Does it increase the uptake with increasing pressure?
So if you just look, so we had also a lot of different pressures and then a lot of different frequencies. And so the way that we analyzed it was based on the initial radius size versus the excursion. And so there was no relationship between the bubble size and the extent of propitium uptake. It was, the only relationship was the shear rate that was produced by a given bubble ultrasound combination and the amount of propitium iodide uptake. So I'm sorry if I'm not answering your question, but we did do some cavitation. We did a small subset of experiments where we did some passive cavitation detection, and we did not find a relationship between stable versus inertial in terms of propensity to cause sonoporation.
So I think we have to go on. Once more, many thanks to the excellent presentation, and I hand over to my co-chair.